Welcome everybody at the final Translational Neuroscience Network webinar of the series 2021-2022. We've had, I think, 8 sessions from all our research programs of AmSam Neuroscience on Translational Neuroscience, and today it's the final one of the season, and we saved the best for last. First, I'll introduce you to Zoom, the platform we're using for this. All the participants from home are not visible. Only the speakers and the hosts will be visible for you. The attendees can pose questions in the Q&A options, and you can also vote for questions that you like, that you think should really be asked. And the chat is not used for the questions. You can choose to adjust the screen, so the slides and the presenters will be in a different format. And welcome. So Translational Neuroscience Network, TN2, has been around for several years now. And initially it was a conference where fundamental scientists and clinical neuroscientists meet to discuss how to translate the findings from the lab to those in the clinic and vice versa. And because of COVID, we started a series of webinars, which were really, well rated and appreciated. And therefore this year we had several based on the research programs. Our focus is neurology, but also psychiatry. And this year we had 9 webinars, and today is the final one. The 9 webinars were focused around the research program, and today the focus is on systems and network neuroscience. And to introduce you the program, I will give the word to Helmut Kessels.
Thank you, Matthijs. Let's see. So, yeah, thank you. This is a theme of the systems and network neuroscience. And in systems and network neuroscience, what we study is, for instance, circuits, how they operate in the brain, and networks, and how they can lead to altered network activity, and also to the disease situations like epilepsy. And also systems neuroscience, in this team of scientists, study how brain regions interact with each other, and maybe also how the brain can interact with other systems in our body. Can I go to the next slide? Thanks very much. So it also says here, and we want to understand how these brain networks give rise to function. And this group, as I said, contains a lot of scientists across all different fields. So both fundamental, preclinical, and clinical scientists. And we are set up to do cross-discipline studies. So the team leaders of this program is both Huib Mansvelder and me. The task force members are Rick Schuurman and Johannes de Boer. And that also leads me to the introduction of the first speaker. I'm very happy that I can introduce Eus, because as a very young person in the 90s, I was still a student. I often like to go to see live music and some of the best bands in the Netherlands. And that's where I met Eus. He didn't saw me, I was in the audience, but fast forward years, and I went to the Netherlands Institute for Neuroscience. And there I met Eus again, which at first confused me a lot. But then I learned that Eus was also not only an excellent musician, but also an excellent scientist. And I got also now to enjoy his science. And I can now say a lot about Eus, for instance, that he published excellent papers, and he's one of the leading figures in the sleep field worldwide. For instance, his group is the one who was the first who used MRI to study sleep, and also set up the Netherlands Sleep Registry. I can also tell you about the great grants he all got for his science, like Vici, an ERC grant, and recently for the second time. But maybe what I like to say is that Eus is also an excellent person, which in the spirit of translational neuroscience, because he also seeks to gather scientists from very fundamental to more applied science to collaborate in sleep. And I also personally know he's also puts in his energy to help younger scientists in their careers. So without further ado, please take the floor.
So thank you, Helmut, for these nice words. I hope you can see the presentation now, right? Okay. So my presentation is called Sleep on It to Ditch or Deepen Distress. So I like to alliterate, hoping that you will remember something about it. Subtitled Why Insomnia is the Primary Modifiable Risk for Emotional Disorders. Of course, this is a very strong statement, but it's also a wish for the fundamental researchers in our great community here in Amsterdam to maybe take on this idea and see what we can do, because I think together with you fundamental researchers, the time is now there to start studying insomnia in, for example, cell culture and animal models. And this has not been possible before. So it's also a call for collaboration with you. What I'm going to present is also collaboration of many people, and I'm very happy to be able to collaborate with all. So you might have to multitask. That's what many people do during a presentation. If this is the only slide you can follow, the whole message is on. You see on the left a sleeping baby. The baby is in REM sleep. We can see that from the twitches in the face and the eye movements. But if you see the mouth moving, this might suggest that there might be emotional things going on during REM sleep. And that's exactly right. The limbic circuit in the brain is very active during REM sleep. And we have the idea that things happen there, that there is a replay of emotional remainders of the day, and that they might either, in good sleeper, facilitate distress, adaptation overnight, or impede it, or even aggravate it. So on the right, you see someone struggling with insomnia. Okay, I'd like to discuss a lot. I will still try to do that in the given time. Just briefly what sleep was again, and then I'll make a statement on why we should study insomnia together with depression and anxiety, so fitting in very much in our program. Some developmental clues, a little bit of explanation of what's actually wrong with our sleep. Then the hypothesis that restless REM sleep plays a key role in disrupting limbic plasticity. Then some inspiration we got from phobia researchers that studied how to get rid of a real bad memory. And then restless REM sleep, how we study that, especially Rick Wassing and my team, using functional MRI and targeted memory reactivation. I'll close with explaining the idea, the hypothesis, with a cartoon, an Engram cartoon. And then I hope to have enough time to show you that these ideas are not just fundamental, but also have applied relevance. We have some nice new results that I would love to share with you if there is enough time. Okay, just briefly a recap for those of you who are not too familiar with sleep. Sleep is an active state, and a lot of replay and plasticity happens. On the upper line, you see from left to right firing neurons, how you might record them in the brain if you could do so during wakefulness, and how on the right that translates to a trace that you can measure on the scalp using EEG recordings like this. Then during sleep, and this is on the bottom left, you see what neuronal firing in the cortex or thalamus might look like during sleep. It seems as if the movie stops all the time, but it continues. So there's alternating a lot of activity of neurons, and then maybe a second or 1.5 seconds, no activity. And that's typical of the deepest sleep. And because all pyramidal cells do that synchronously over the cortex, you can record these huge waves going on. We call them slow waves or delta waves. So that is the recap. This is 20 seconds here. So if you would have to record a full night, that's not very handy. So what we usually do is we have all these 20-second epochs, and then we just give a representation like this here from about 11 at night to 5 in the morning. If we record wakefulness, we set a tick mark here, and then people go into stage 1, 2, and 3 sleep. This is the slow waves here. Then after about 1.5 hour, people might go into REM sleep in red here. So this is just a way of visualizing a whole night to have a glance at what people do across the night. And I will explain this because I will get back to this. So I study insomnia, and I really want to draw your attention to that it's not trivial to study insomnia, because there are very strong socio-economic and clinical bottlenecks to solve. And I think we are in an excellent position here in Amsterdam to do so. So if we think of mental disorders, we know that there are soaring costs of burden, and that anxiety, insomnia, and depression are the top 3 most common. So they account for 2/3 of all mentally affected people in Europe. Oftentimes, insomnia is forgotten for some reason, but we have to include them. And we see anxiety disorders together are the largest part, then insomnia, and only then bipolar depression, and only then things that we also like to study, like dementia, ADHD, etc. Now, if we look at the number 1 risk for anxiety or depression, if it's studied well, you always find that insomnia seems to be a key factor. Now, the problem with these 3 types of disorders is that the treatment is very separate. There is limited efficacy, and the mechanisms are not really known. Also not how they connect together. And this is something I really want to work on and continue working on. I was fortunate enough to get another ERC Advanced grant to do so. And what I want to do in that Advanced grant is break the wall between insomnia research and research on anxiety and depressions. And I think it's relevant to do so and necessary to do so. Because in insomnia, we have this high prevalence of anxiety and depression. In people with anxiety and depression, I think 4 out of 5 also have clinically relevant insomnia. Then a key symptom of insomnia is hyperarousal, and I will get back to that, what it means. So hyperarousal determines the risk, the severity of the insomnia, and also the risk of relapse. And it turns out to be about the same in many people with anxiety disorders and depression. So hyperarousal is key to these disorders. And then with the great work of Daniel Postma's team, we found that if you look at genetic correlations of the risk of insomnia, they rank highest with the risk of anxiety and depression, either symptoms or diagnosis. So there's something there that links these disorders. And there's another observation. We know that childhood trauma plays a big role in all these disorders. And if you look at the effect of childhood trauma on the development of anxiety and depression, it seems mediated through insomnia, as if trauma puts you at risk of insomnia and then developing anxiety and depression. So this is what I'd like to do in my next 5 years, try to integrate these approaches, bring down the wall, focus on hyperarousal, the mechanisms underlying that. And the way I will do it is trying to link 2 somewhat separated fields. So those working in sleep know that during the last 20 years, there have been thousands of papers showing that during sleep, we do things like learning and forgetting stuff. Now, people not working on sleep, but on emotion regulation, they are very much focused on adapting to emotional distress. How do people become sensitized, and the other way around, how can they adapt to distress? But if you think of it, the bottom line of these very separate fields is basically in the brain about the same. It's all about neuronal network plasticity, changing connections, like here visualized, in neuronal networks. That holds for learning and forgetting during sleep, but also for adapting to emotional distress. And I think it's very valuable to try to connect these now quite separate areas. So just a brief word on how people develop a vulnerability to develop insomnia later in life. Well, genes and early life stress contribute to the risk of this baby that you've seen before to develop into that vulnerable adult that you have seen before as well. These are two important factors in it. We know that the heritability is for insomnia is quite high, it's 40%, and that there is, I mentioned already, the high genetic correlation with anxiety and depression risk. And somewhat surprising, and that's also the work of Daniel Postma's team, these insomnia genes, we did not really find them a lot in sleep regulating structures, but they were very common in emotion regulating circuits. And I think if you want to remember another thing from the talk, That is maybe that insomnia, we see it as a sleep disorder, but maybe it's an emotion regulation disorder. That childhood adversity is also very interesting because we have seen that childhood adversity, not in our team, but in other researchers, increases arousals from REM sleep in adulthood, as if it's more difficult for people to maintain REM sleep and they're more likely to leave that before necessary. And another thing I think is very common in people with insomnia, they're so good at staying watchful. And if you think of children at risk of neglect and sexual harassment and violence, of course, that's in a period of time that the brain is very plastic. So their brain may have been developed to optimally stay watchful all the time. So that may come in handy at that time, but it's not very useful if you want to go for restful sleep. Okay, zooming in on what is wrong with the sleep of people with insomnia, but often found also in anxiety, depression, and post-traumatic stress disorder. It is not so much that they do not sleep, because here on top you see a few hours of sleep of a control sleeper, a good sleeper, and here someone could be one of these disorders. There is red, green, and blue indicating that there is sleep. Here you see that for these patients, there is sleep. But the thing that's most remarked is that they leave sleep all the time for either other sleep stages or wakefulness, very brief arousals like shown here, just a few seconds, or full 20-second epochs of wakefulness. So it's the fragmentation, it's the barcode-like profile. If you compare, for example, REM sleep of a good sleeper, it's very consolidated. It is very fragmented. And that made us think, well, maybe it's the fragmentation that does the harm. So what is the harm? For a good sleeper, it's even a saying, sleeping on it, or een nachtje overslapen in Dutch. Usually if you're very distressed or sad or anxious or whatever, if you have a good night of sleep, usually you wake up feeling much better. And it's common for people with insomnia, anxiety, depression, and PTSD not to wake up feeling better, but actually sometimes even to wake up worse than they got to sleep. And this, I think I interpreted this as a clue to maybe there is something going wrong in overnight emotional memory trace plasticity. So why would you make things worse overnight instead of alleviating the stress? So this is the hypothesis, and we focused on restless REM sleep because the limbic circuit is so active then. We know that a lot is happening during sleep, things like memory consolidation, adaptation of memory traces or engrams, how you want to call it, and also dissolving of emotional distress. That for the brain, these processes, as I mentioned, they are all neuronal plasticity and changing synaptic strength in networks. So that's what we focused on. So why REM sleep? Well, we found that REM sleep is a very special time for neuronal plasticity because this is very old recordings by Gary Aston Jones about 40 years ago. This is multiple unit activity here of the locus coeruleus. And you see that's going on basically all the time. This is EEG of the rat, and when the amplitude is high, the rat is in deep sleep. And this is EMG, and then you see that both during deep sleep and wakefulness, the locus goes on and on. So meaning that there will be adrenaline. And only if the rat goes into REM sleep, here, there is this complete absence of locus activity. This has been replicated with nicer tools just recently by Cecilia Kjerby, Mike and Nedergaard's group. So here they recorded simultaneously in mice a lot of things, so they could measure activity of locus neurons. And REM sleep is here, these greenish bouts, I colored them here. And they recorded simultaneously the amount of adrenaline in the prefrontal cortex. And you see that if REM sleep starts, and only in REM sleep, you have this slow time constant decrease in noradrenaline in the cortex. But as soon as there is an arousal, it can be very brief, noradrenaline shoots up. So you need long consolidated sleep to reach low levels of noradrenaline in the cortex. And also, of course, in other places where LC projects. Now, and if you think of that this might happen in good sleepers, so that noradrenaline may be high in waking, a bit low in slow, but only during REM sleep, really absent. But not in bad sleepers, because they shoot out of REM sleep all the time, so they never reach this low level of noradrenaline. So that's the hypothesis. Of course, in humans, this will be very difficult to record, but at least in animals, there is support of this. Now, why would it be so interesting to have low levels of noradrenaline for a while? And this is something I got the idea from Miro Kind, basically, and Joe Ledoux earlier. And I'll walk you through. So I switched now to people that treat phobia, and they found a way that helps in some people. Well, suppose that you meet this rabid dog and it's barking at you. There is something happening, and whatever your brain was doing, the locus kicks in, releases noradrenaline, and all the circuits that were active stop working, and the priority is given to limbic and salient circuits. So that's what noradrenaline does for you, and fortunately so. Now, people may have develop a dysfunctional association, where if they see this fluffy, nice animal and say, what kind of nice thing is that? And then they realize it's a dog, and they are, for example, phobic about dogs, immediately they get this fear response. No need for that, but they can't help it. Now, the thing is, the idea is that if there is this fear response for a brief while, there is a lability of the synapse. And in the studies that have been successful in some people, if you then induce fear in these people, give them propranolol to block the effects of noradrenaline that increases, then it may happen in people that somehow, if they're presented with a dog again, they don't have that dysfunctional association anymore, as if the synapse has been depotentiated, or the link has been broken. But this gave us the idea, and also based on studies of animal researchers like Gina Poe and also Helmut Kessels, that if you have REM sleep, and there is an animal model for that is restful, there can be a balance between long-term potentiation and depotentiation, and fear might extinguish overnight. Now, in these very stressed rats, in the model of Gina Poe, for example, there is restless REM sleep instead, like in the insomniacs, and maybe that balance of depotentiation and potentiation is disturbed, and there may even be fear enhancement across sleep. This is just something we're working on. So this is the idea. If there is activity in networks in the absence of noradrenaline, you might favor long-term depression. If there is the same activity in the circuit, but there is high noradrenaline, you might favor long-term potentiation. And these are a few examples of papers from very basic processes until really learning overnight, and damaged learning if you mess around with locus activity. So this is, I think, very relevant, especially for emotional processes. Why? And that is because in REM sleep, these limbic circuits like anterior cingulate, amygdala, insular cortex, orbital frontal cortex, they become very active, even more active than during wakefulness. So if there is replay of memory traces, they will include these parts. So this is the idea. If there is restless REM sleep in people with insomnia, they will never reach these low levels of noradrenaline, while good sleepers may do so. So how could we study that? And I now turn into the work that Rick Wassing has been doing. This is what the idea was. We give people a stressor, then we do that in the scanner. We can, for example, record amygdala activity. They have good sleep, we record their sleep. We give the stressor again the next day, and we see adaptation of the amygdala. That's the hypothesis. And if we do that in people with this fragmented sleep that never reach that low level of noradrenaline overnight, the hypothesis, there will not be this decrease in activation of the limbic circuit if we give the stressor again in the morning. Then the question is, so this is what we want to do, give a stressor and give a stressor again after sleep, after consolidated sleep or disrupted sleep. But what stressor should it be? And this is something where I took my experience in music. And this was the protocol we developed. So we recorded people singing, wearing big muffles over their ear, headphones. So they only heard other people singing and not very well their own voice. So they got out of tune. And I'll show you, we recorded that. This is Rick singing. I instructed him to destroy my recordings.
Stille Nacht, heilige Nacht, Davids Zoon, lang verwacht.
So I hope you heard it. So listening to your own voice can be embarrassing, and listening to your own voice singing out of tune is really embarrassing for many people. They really feel the stress in a physical way, like sweating hands or pounding heart or weak knees or feeling nausea. So this is what we wanted to accomplish, and we tested that. And this was the first test, just recording physical signs of distress, of shame. And these were good sleepers. So overnight, it was less embarrassing for them. They had less of these physical symptoms if they listened to them. And this is insomniacs here. And we thought it would be a flat line, so they would have no benefit of sleep. But sleep, as you see, even made things worse. So sensitization instead of adaptation. So we took the protocol to the scanner, and then we indeed found that the more time people spent in REM sleep, the more overnight decrease in amygdala activation upon replay of that embarrassing experience. But the kicker was that people that had minimal rest in interrupted REM sleep indeed had a strong decrease. This is more to the left, is a stronger decrease with good sleep. But the people that had fragmented REM sleep, they even moved to the other side of the line. So some people ending up with sensitizing the amygdala overnight rather than desensitizing. And we had a targeted memory reactivation protocol adding to it to show that there was some, really some causality in it. We could boost both ways, either sensitization or adaptation. So I want to close with a cartoon. I'm over time already, but I think it's a nice way to remember it. Suppose you experience a real bad accident. This morning, many people, experience then physical signs, like in the picture here. That's all part of the limbic system. It records it, generates it. In the meantime, you see and hear the accident. So you have this link here in purple between the facts and the arousal that you can feel in your body. Many people know that if later on the day you tell this experience to someone, you could feel again that physical distress in your body. You might feel your heart pounding or the weak knees or whatever again. But just talking about the facts, you trigger that arousal. Now, replay during the night, at least in good sleepers, seems to do something. If there is low noradrenaline during REM sleep, the next day, if you talk about it or you recall the accident, you can do so without the arousal, the hyperarousal in your body. And the idea is that people with this disrupted sleep, where there is no low levels of noradrenaline ever, that breaking of these purple links fails, and they end up, if they recall whatever context, everything starts to generate that arousal. So with that cartoon, I would like to close and thank all my collaborators, and sorry to be over time.
Thank you. Thank you, Eus, for an excellent presentation. I learned a lot. I'm not very familiar with all the network sciences, but it was really informative. If I may start with a question myself, do you you also have excessive sleepers? Can you learn something from them to understand the mechanisms in insomnia and see whether excessive sleep is like the opposite phenotype?
We have too few recordings ourselves to do that. There are some excessive sleepers where sleep is still fragmented. For example, some people with sleep apnea, they have a choice of either sleeping or breathing, So of course the brainstem kicks in and wakes them up all the time, so they have fragmented sleep as well. They also are at higher risk of, especially that group, of developing depression, for example. But these people might have really long nights and they say, I can sleep for 10 hours and still feel that I need 2 more hours of sleep. But there are also people who have hypersomnia, and yeah, but we don't know what's happening there. Narcolepsy is another example. They have hypersomnia, but they also have very fragmented sleep. So it's difficult to learn from those cases.
It's more about quality of sleep then. Okay. There were some questions from in the chat, Helmut.
Wait, I gotta unmute. Sorry. Thanks, yes, Matthijs. There's a question from Bert de Dacht. He asks, which proportion of the genetic associations with insomnia is epigenetically programmed?
I don't know. I really don't know. So everything we found is in the GWAS we found associations. It's like usually it's only small percentages, and then what happens epigenetically, I don't know. I'm sorry.
Could we say that if early life stress would have a big effect, you could call that epigenetic?
I think we can do, but we don't know the real link. So the link that I could find, and I think is relevant for the idea, is that we can record restless REM sleep in adults that have been exposed to childhood trauma. So that's something that sort of supports the idea. But then the what's happening epigenetically and in which circuits and why then they develop restless REM sleep, we really don't know. I see that there is another question. I don't know if there is time.
Yeah.
Yes.
The other question is meditation, half an hour a day, not thinking, a good treatment for insomnia?
Mixed findings. So it's not the best treatment for sure. So many people, I have an example in the family, they just cannot relax. They cannot shed the hyperarousal. So they become tense of even trying to meditate. So the best way to handle insomnia, not always solve insomnia, is to do cognitive behavior therapy. And the most effective component there is shortening sleep. So people are allowed to be in bed only for a short time, maybe 6.5 hours. And what that does is it compresses sleep and makes it less restless. And Rick has been following up his studies in Sydney now, and he showed that if you compress sleep to a shorter time window, that restless REM sleep indeed decreases a lot. And meditation, I think, if you also look at what are effective treatments for depression, I don't think meditation and mindfulness and things like that are too effective compared to the other things like activation.
All right. Another says, thank you for the excellent talk. Can sleep stages be focal to a specific brain area? For example, only few brain areas can go into REM, or is it always a global phenomenon?
I think the sleep stages are global. That means that in every stage, a lot changes. So as I mentioned, and I think it's most important, REM sleep is very special because it's the only time when the locus is really, really suppressed. And in the meantime, the limbic system is extremely active, even more active than during wakefulness. And during other stages, there are other constellations of activity, higher and lower in different brain areas.
And another question is MDD, in MDD, there's often an increase in REM quantity. Would you explain this as the body trying to more successfully process emotions since the higher fragmentation rate?
This has been a hypothesis. I think, so there are some things that might seem conflicting, because if you have an antidepressant, it doesn't matter what, either a pill or ECT or whatever, if the treatment is effective, usually you see that it suppresses REM sleep a lot. So then that seems to be conflicting with a positive role of REM sleep. But I think this is the answer to it. You'd better have no REM sleep than fragmented REM sleep. And then ideally you have consolidated REM sleep. Because if you have fragmented REM sleep, our observations suggest that there may be sensitization overnight of bad things rather than desensitization.
So I think we have to proceed to the next speaker. Thank you very much, Eus. And at the end of the session, we will have a moment with the host and both speakers to go into any remaining questions. But right now, I'd like to continue by introducing the second speaker, Willem Dahan, who's a neurologist at Amsterdam UMC and works as a postdoctoral researcher at the VUMC Alzheimer Center. He did his PhD on functional brain network changes in Alzheimer's disease and continued his research on the neurophysiology of dementia. His projects mainly focus on fundamental studies on magnetic encephalography and EEG, network analysis, computational modeling, and non-invasive brain stimulation. And he also developed brain visualization software and heads the EEG lab in the VUMC. And well, we're looking forward to your talk, Willem.
Thank you, Matthijs. And let me share my screen. Hopefully you see the first slide now on your screen. Well, hello, everyone. My name is Willem Dahan, a neurologist at the VU Hospital, and mainly work at the Alzheimer's Center. And for research, my main focus is on the clinical neurophysiology of dementia. And what I would like to show today is that development that of the previous years is that we have been starting to use computational models in our research. And we think this is can be a very powerful and interesting combination with neurophysiology. And this started out with having a tool to try to better understand the data, our patients' data, and the changes that we see happening in Alzheimer's disease. But we gradually also moved towards actually intervening in the model and maybe even seeing if you can do something like treatment prediction. And those are things I would like to talk about today. So to dive straight into a one-slide summary of neurophysiology in Alzheimer's disease. This is what you see now is actually a bit outside my own field, but if you look more at the microscopic or the cellular level, an insight of recent years has been that the amyloid plaque or the amyloid deposition, one of the pathological hallmarks of AD, gives rise to pronounced hyperactivity and hyperexcitability of the neurons. And this has led many authors to say that we should actually maybe focus more on that and maybe even as an alternative type of treatment, trying to target this or regulate this hyperexcitability. So that's something I will come back to later. It's more nuanced than this because there's also tau, and tau might actually have a hypoactive effect, and there might be some kind of balance between them. But this hyperexcitability is something that is now quite well established. So when we look at our data, this is mainly EEG and MEG data, which is at a much larger level, of course, is brain-wide activity recordings in humans. The question is, do we see signs of this hyperactivity and hyperexcitability? And well, the answer is no, because what is the typical finding in Alzheimer's patients is that you see a gradual slowing of oscillatory activity. So the human alpha rhythm or the posterior dominant rhythm slows down, and we see more slower frequencies like theta power. Then if you go into more advanced analysis, looking at communication between brain regions or even network analysis, it's mainly a story of loss. So you have a loss of a weakening of functional connections, and there's a loss of network structure and hierarchy. And one interesting phenomenon is that mainly these highly connected functional hub regions, they are somehow selectively vulnerable in Alzheimer's disease. And that's also something I'll get back to later. But there's a discrepancy here. And of course, these findings are all probably part of one disease mechanism that we ultimately, of course, would like to have a coherent multi-scale view of. So the question is, how could you achieve that? And experimentally, if you think about it, what kind of experiments you would have to set up if you that would require invasive recordings in AD patients in a lot of different brain regions, maybe even repeated or longitudinal, and combined maybe with large scale macroscopic recording. So that becomes a setup that's not feasible and not that's too challenging and too burdensome in AD patients. So yeah, the question is there an alternative? And that is where modeling comes in. So here you see a very short fragment of EEG data, and yeah, this is just raw data, 20 channel resting state, broadband data. And let me just quickly stop the animation for a second, because I noticed that the previous time I started, the program crashed. So brain dynamics is a bit too much for PowerPoint to handle. But what I want to point out is that looking at this in a 3D visualization is nice. But the question, of course, is, does it help you to understand what's going on inside? And yeah, of course, we have a lot of quantitative measures also to look at activity and describe local activity and connectivity. But yeah, what I would like to point out is that because of the enormous complexity of brain dynamics, the multi-scale nature, the hierarchy, the nonlinear effects, we really need more sophisticated models to try to find coordinating principles. And in fact, if you look at other complex system fields, for example, telecommunication or economy, there also the use of models to understand and predict effects is very common practice and often essential. So yeah, so the complexity is one argument to use modeling to make sense of the data, and the technical limits I talked about before. It's very hard probably to do everything experimentally. Another thing why we think this is really a nice complementary way of looking at data next to our patient data is because you could regard looking at the patient data more as a top-down process where you try to understand and interpret your findings, whereas here in a model you can build it and you can simulate the changes that you see in the patient data, or you can manipulate them, and you can even maybe try to counter them and go towards a remedy for changes that you see. And that's something I will talk about later as well. So it's good to realize that computational modeling in neurophysiology is not new. And it's also good to realize that there are different scales on which you can work. And there have been in previous decades already a lot of very successful computational models, mainly at the lower scale, so at cellular or even subcellular levels, going all the way to like ion channels, but also neuronal circuits, so smaller groups of neurons and how they interact. And I think a more recent development is the larger scale macroscopic brain network simulations. One of the more well-known packages, for example, is the virtual brain. So yeah, ultimately, you also would like to link these different levels, of course, or have a model that merges these types of simulations. And I would like to go into a little bit more detail about the model that we prefer and that we have been working with. So these are based on so-called neural mass models, which have been around already also for quite some time. This is one based on work by Lopez da Silva. And what you see here is actually a description of a neural mass, which is basically a set of differential equations that describes the activity in large groups of neurons. And well, the abbreviations here are not, I don't have the legend here, but these are parameters that describe relevant characteristics like membrane potential or spiking rate, coupling strength, and so on. And if you implement a model like this in software, you can actually run it, and it generates a fairly realistic human alpha rhythm. That's where this model was developed for. So yeah, this simulates activity of a smaller brain region. Of course, we would like to go to brain-wide networks. So in the previous years, what Professor Kees Stam from our department has been developing is a software called Brainwave, where we take these neural masses and where we couple a large number of them, for example, 80 of these regions, And we couple them according to what we know about human topology or human coupling between brain regions, for example, based on diffusion tensor imaging data. So then you end up with a network that you can run, and it generates EEG or MEG-like activity that you can also compare, so that you can compare the output to patient data. And the nice thing, of course, is that you can influence the model. So you can turn the knobs and you can, for example, change excitability or coupling and see then what the effect is at a larger scale. So what I would like to show is 4 different examples of things that you can do with a model like that. And that's moving from where we started trying to understand the changes in our patient data more towards can we try to link different scales? And then finally more towards can we also somehow change back this damage that we see, and can we counter the slowing and the loss of functional connectivity, and can we use that actually to inform, go back to the patients again and inform actual empirical experiments. So starting with trying to develop some kind of damage model of neurophysiological damage model, we were very fascinated by this phenomenon that in our data and also in using, for example, fMRI, there seemed to be in Alzheimer this selective vulnerability of these highly connected hub regions. And we were wondering why, and well, one obvious answer could be, well, maybe these highly connected regions, because they're highly connected, on average, they're also more active, and this chronic excessive level of activity makes them more prone to degeneration. We also know that in these hub areas, there's more AD pathology, and well, we know the effect of amyloid leading to different levels of activity. So, and there are other factors that might play a role here. So we thought maybe we can implement some kind of damage rule in our simulation model, where we say, okay, we are going to damage the physical network based on the level of activity of a certain region. So the more active a region, the more damage it will receive. And then the question was, does this lead to macroscopic oscillatory changes that we also see in Alzheimer patients? And this was indeed the case. So we were able to, using this, reproduce the typical slowing and the loss of connectivity, which I'm not showing here. But what I am showing here is something that kind of surprised us because we actually, from the model, you can also read out things like spike density, so firing rates. This is not possible from our patient data, but we can see this in the model. And there we saw this early transient phase of hyperactivity, and this was more strong in the hubs. And this was surprising to us, but when we then started looking at the more fundamental literature, these stories of hyperactivity, we thought, hey, this model that we have here, or this damage rule, might actually be a good approximation of what happens in Alzheimer's disease, and maybe we can use this to do more experiments with. So this was a fairly simple damage rule. We then also thought maybe, because there's much more knowledge on the microscopic level, of course, about what actually happens due to, for example, amyloid or other factors. And we know that there's interneuron dysfunction, specifically from the lab from Ronald van Kesteren, also, for example, who had his inaugural lecture yesterday, and we collaborate with him on this. So there's quite some knowledge about what happens to neuronal circuits. There's also things like glutamate toxicity and other mechanisms. So we try to look at our neural mass model and see if it's possible to make a more direct implementation or change in the model that reflects these specific mechanisms. And up to a certain degree, we were able to choose parameters for that to reflect these different types of changes. And then again, the question is, what happens at the larger scale? Do we see changes of the brain network that we see in Alzheimer patients? So this has been mainly work from our PhD student, Anne van Nifterik, who has done really an extensive search on all kinds of hyperactivity regimes that we could think of. And while there's a lot of information on this slide, it's not about the details, but what I would like to point out is that if you see the gray area, so these are all the occasions where the different regimes actually resembled the patient data. So I think it's clear that the AD-like scenarios, so the scenarios of hyperactivity, they actually resembled led to oscillatory changes that resemble what we see in AD patients, so slowing. And yeah, this still feels counterintuitive, but here she showed that actually lower level hyperactivity is in fact compatible with oscillatory slowing at the larger scale. So zooming out a bit, and this is part of a recent review where we also argued for the more prominent role of neurophysiology in AD research. If you see that, yeah, there's a lot of knowledge on neurophysiological changes in AD at different levels, and there are good techniques to describe them. Yeah, the idea is that the model, and there have been already a lot of adequate models at different levels. Can we use this type of modeling to actually merge these different levels? And yeah, we think that this is a potentially rewarding way forward. And now I've talked about neural mass modeling, but there are also other types of models where, for example, that focus on spread of pathology across the brain, or models that take into account plasticity, for example, a very important aspect which we have not really gotten into yet. So moving on to if you are able to kind of describe the damage in a network that you see, and maybe principles of how this damage occurs, can you also reverse it and maybe change the model or intervene in the model and find ways to counter the changes and get activity back to more normal levels, so higher peak frequencies or higher faster oscillations, for example, resembling what has been shown already much earlier with the use of cholinesterase inhibitors, so the medication that we have at the moment, which studies show that they were able to kind of temporarily reverse the slowing that has been seen. So in the neural mass models, there are the different groups of neurons, the excitatory and the inhibitory groups. So we started out with a few of global strategies where we say, okay, we're just going to change the level of the excitability of certain neuron groups and then see what happens to the network activity. And you could do that at a global level, so change the groups together, or just selectively change, for example, only the inhibitory interneurons, for example. And this led to 6 different strategies. And, well, this is not a very quantitative output, but it's just a short overview where we compared these different strategies. And yeah, so their performance and their success was based on how long they were able to keep the network activity within a normal range. So we ran the simulation, there was the damage, the Alzheimer damage applied, and then simultaneously we started one of these strategies to see if we could slow down the detrimental effect on the network. And here the stimulation of excitatory neurons strategy seemed to be the most successful. Yeah, this was a start to try to use modeling for this purpose. We actually want to become more specific with this and also really try to move towards actual experiments. So a project that is running now, and this is mainly the work of PhD student Janne Lupi, is where we start to use a method of non-invasive brain stimulation. And we use transcranial direct current stimulation, or TDCS, which is a very patient-friendly technique in the sense that it's not very burdensome. There are virtually no side effects except for some slight tingling. And there has been, in previous years, there have been some positive reports, positive effects on cognition. Although at this point, I should say, I think the literature is still very inconsistent. And often, yeah, the outcome measures are cognition. So it's not very clear what the mechanism is that how these DCS or alternating current stimulation, how it's actually able to improve cognition. So this is something we would like to study. And the great advantage of DCS is that we can also use this in combination with our MEG system. So we can, while we stimulate, we can also record brain activity at the same time. So we can monitor if what we think we are doing with stimulation, if we're actually achieving that, or if completely other things happen. And yeah, we also, of course, if you start stimulation experiments, the question still is, okay, where do you want to stimulate on the scalp, how strong, and so on. So that's also, therefore, we also want to still use the modeling, because it's possible to simulate DCS stimulation in the models with a technique, for example, like current flow modeling. You can, if you have a certain location of your stimulator, a certain strength, you can calculate which underlying regions will be influenced. And you can use that in your simulation to test different positions, different strategies, and yeah, make a virtual prediction of what could, in theory, be a good way to perform an actual experiment. So this is a modeling phase that we're in at the moment. So we're trying to find virtually the best options, the best stimulation regimes. But then, of course, in the end, you have to test this with actual experiments. And that is something that we'll start to run next of this year, actually. And very anxious to see if our predictions will actually also hold. And I think it's good to say here that at the moment we're not aiming for substantial cognitive improvement, but we're really looking at the short-term effects, so short-term change of the network activity in the right direction. This is a bit more about the workflow that we're looking at, but I think looking at the time, maybe for now, this is not, I will not go into detail about this, but it's just we're trying to, the bottom line is we're trying to bring the modeling and the actual experiments closer. So wrapping up, I hope to have shown you a bit that the combination of neurophysiology and computational modeling is interesting. And there's a lot of different things you can do with it. And in fact, yeah, especially the neural mass models are, I think, very relevant. There are also a few caveats, of course, and most people who work with models know this, is that it's easy to get carried away and try to add a lot of realism to your model, but this also exponentially increases the complexity of your model, and also the interpretability, decreases the interpretability of your model. So you still need a good research question, a good hypothesis, and then pick the right model for that, and maybe even constrain the model to get an outcome that you can also are able to understand. And of course, what still needs to be done much more is that we, besides all this modeling, we need to validate the models with, well, on the lower level with the animal experiment data, and on the large scale level with human data. Yeah, so that's what we will be working on for the coming time. Yeah, on behalf of our clinical neurophysiology of dementia group, thanks for your attention, and I'll be happy to take any questions.
Well, thanks a lot, Willem. I really learned a lot again. So anyone, if you have any questions, please put them in the Q&A box. I'd like to start off with a question, because I think you've developed this excellent techniques in several ways of measuring and modeling. So is it something that you can apply generically to a lot of neurodegenerative diseases like Parkinson's disease, ALS, or is it like a method that's easy applicable to all kind of brain disease?
Yeah, great question. Actually, something I wanted to mention, but forgot. But definitely, I think this is, of course, I'm mainly focused on Alzheimer's disease, but in our group, we have also used the similar type of neural mass models, for example, to look at epilepsy or more general network effects. But I think you can really expand this to a lot of other types of diseases, other neurodegenerative or other types of neurological diseases. Definitely, yeah. It's a very general way of looking at brain activity.
Yeah. So there is a question. Is it possible to selectively target excitatory or inhibitory networks using TDCS?
Using TDCS, yeah, that's a good question. If you look into the TDCS literature, I think there's actually quite some conflicting information about the presumed specific effect of the stimulation. People saying, well, probably it's mainly and I think this is the consensus, that you mainly influence the excitatories or the pyramidal neurons. But there are also people say, well, it really depends on the region, and you might also have a more strong effect on the inhibitory interneurons. So there are it's a bit conflicting views. I don't think, also because of the changing orientation, because of the cortex, you can become very specific about stimulating specific circuits. So I think, yeah, there are high density techniques that are able to focus on a smaller spatial region, but I don't think you can get very selective in the type of tissue that you want to influence. But yeah, there's still a lot of variables, because you can change the position and the intensity and the duration. So yeah, that's also a reason why we want to use the modeling and the actual experiments to just try to monitor what actually happens when you pick a certain region, and maybe the modeling can also help us to find out actually what the stimulation at a certain place is doing, and which of these groups, so the excitatory or inhibitory, are actually influenced the most. So maybe it can help us in this sense as well. Yeah.
Yeah. So I was thinking about this stimulation. Is that something that has also already been done in animal experiments, maybe to show how you can influence these networks?
You mean like the electrical stimulation?
Yeah.
Yeah, there are examples of that, and that's also something that we are with a consortium are planning to do. Is to there have been, for example, interesting results of 40 hertz gamma stimulation with different modalities, but also electrical, for example, with alternating current, and both in humans and in animal experiments. And there, of course, in the animal experiments, you are more able to record invasively and study the mechanism with more detail. I don't think at the moment we can say that we understand the mechanism, but I think this is something that will come up in the coming years, that because of the initial positive reports on cognition, we really now would like to better understand the mechanism and see how we can optimize it. Yeah.
There's also another question, Willem, in the Q&A from Klaus. Which oscillation properties are used to tune the neural mass models? And did you check whether oscillations exhibit long-range temporal correlation?
Yeah, so what the model we use most, they are based on the, like I told about, the Lopez da Silva neural mass model, which was primarily developed to focus on replicating or simulating the human alpha rhythm. So there are different parameters that I did not go into detail now, have been tuned to get as realistically possible alpha rhythm oscillations when you run that model. Of course, that model, for example, is focused mainly on the alpha rhythm, and it does not really produce the full typical 1/f spectrum that you see in human data. But you can, there are other versions of this type of model that add, for example, other inhibitory or excitatory groups or other loops that help to get a more rich spectrum of oscillation. So I think, yeah, you can tune the model according to a lot of different parameters. And I think referring to the second question here, I think we have not really looked specifically at long-term autocorrelations, if I remember correctly. But it's something that you could use it for in principle, I guess. Yeah.
Okay, thanks, Willem. So I think we're at the end of the second session. I'd like to invite Eus back to the floor. There are some compliments that the talks were very informative, and there's a leftover question for Eus. There were from UXC, she describes she had episodes of insomnia with hyperarousal. But if I did some mild running before sleeping, I would experience much less hyperarousal and much easier in falling asleep. Can you explain the change with your proposed model?
Yeah, so it's something that very early in my scientific career we studied this also, that and it has been replicated several times. Strong exercise can boost sleep pressure. So if you boost sleep pressure, as is also done in, for example, curtailing sleep in people with insomnia with cognitive behavior therapy, so anything you do to boost sleep pressure can make sleep a bit better. So that's how it fits in the model.
Okay, thanks. And Willem, I was just wondering, maybe it's a strange idea, but if you would use the transcranial stimulation on healthy persons, could it improve memory?
Yeah, well, yeah, well, a lot of people are also trying to look into this, of course, and I think there are even groups or maybe like even companies claiming that they are able to do this. I have actually heard anecdotes of patients who were approached to receive sessions for dementia. So this is not healthy persons, but patients. But I know it's also occurring in healthy persons. Yeah, where they actually offered multi-session therapy for quite large amounts to boost memory. But yeah, I think at the moment it's honest to say that we really do not yet know how to do this successfully. Yeah, so also for improving healthy cognition. Yeah, at the moment, I think I hopefully with exploring the options systematically, like we try to do with the modeling, that can help to look at these effects. But yeah, at the moment, I don't think we can say that it's possible yet. Yeah.
Helmut, any residual questions from your side?
I was wondering, a question for Willem. You said you are going to select patients for the trial. I was curious what your selection criteria were, because you probably want patients that have increased network activity.
Yeah, so this is a good question. So, in what phase of the disease would you like to include patients? And for now, here we chose to recruit people who are in the early phase of the dementia phase, in the early stage of the dementia phase. So they are still able to, and these are often participants also for clinical trials, for example, because they are still able to oversee their contribution or decide on whether they would like to join. But when we do EEG or MEG recordings, we do see clear changes in their brain activity. So we have something to actually improve on. So well, that's a start for us. I think going towards maybe even earlier phases, so people who are not in the dementia phase yet, but where we already know that the Alzheimer pathology is present and who have cognitive complaints, that could also be an interesting group. But we started with the early phase dementia group.
Okay, well, so again, I'd like to thank our speakers, Willem and Eus, for their excellent presentations to give us some view of what richness there is in the systems neurology network. And with that, I'd like to close and thank all the attendees for joining the session.